Hello everyone, and thank you for joining today's session. I'm Shawn Sternisha, the Commercial Product Manager for our ultracentrifugation portfolio at Beckman Coulter, and I'm joined today by my colleague Bala Venkatakrishnan from our applications team. We're excited to walk you through how the OptiMATE Gradient Maker enables scalable and reproducible ultracentrifugation through the automated preparation of density gradients. Before we dive in, just a quick note here on regulatory language. The protocols and workflows that we'll discuss today are intended for research use only and are not validated for diagnostic applications. Please also be aware that the content is proprietary to Beckman Coulter and is not to be redistributed or copied without permission. So let's jump in. We can begin by thinking about the why. So density gradient ultracentrifugation, or DGUC, has long been a gold standard for difficult separations that require both high purity and yield. It's been used in a wide variety of applications, including viruses, which is primarily what we'll focus on today, but it's also been used in nucleic acids, where it enables the separation of various forms of plasmids, and in extracellular vesicles, where density gradients allow for the isolation of specific subpopulations. And there are numerous other applications, including cells, organelles, protein complexes, and more. But regardless of what you're purifying, density gradients often provide unmatched resolution, especially when you're trying to separate closely related populations, so things like empty and full AAV capsids. And in many cases, density gradient ultracentrifugation is the only universal purification method. For example, with AAV, it can work across all serotypes without additional optimization being required. So one important consideration is the type of density gradient being used, and your choice will depend on what you're trying to separate. So starting on the left side here, I'm sure everyone is familiar with pelleting. And this is only included here as a means for comparison, since it's not a density gradient method. Much like pelleting, however, rate zonal centrifugation separates also on the basis of sedimentation coefficient, or S value, but it does so with significantly higher resolution than pelleting. And in contrast, rate zonal relies on a preformed linear density gradient, which modulates the velocity at which the sample components will migrate to the bottom of the tube. And so therefore this is a time-resolved technique. Rate zonal has traditionally been somewhat cumbersome just due to difficulties forming the linear gradient manually. The next approach here is called isopycnic, and in this case, separation occurs due to subtle differences in the buoyant densities of the particles, so not their S value. And although linear gradients are also used in this case, the starting point of the separation is quite different from rate zonal, and traditionally it's beginning with a homogeneous solution of your sample mixed with a gradient forming material. So the gradient here is not formed manually, but rather it only forms once centrifugal force is applied, and this is an equilibrium approach. Lastly, in the far right column, equilibrium zonal is yet another method whereby the particles are separated by their buoyant density, but in this case, discrete steps or layers are used. So you're not forming a linear gradient, and therefore the resolution is not quite as high as isopycnic, but because you're not spending that time to wait for the gradient to form, these experiments are often much quicker. And as you'll see later, OptiMATE can dispense both step and preformed linear gradients, so it can enable all of these methods. As I'm sure many of you know, traditional density gradient workflows have not always been easy. When it comes to preparing the tubes, for example, it's rather labor intensive, especially if you need to begin with a linear gradient, or if you have to pipette many distinct layers. Because of this manual nature, variability is a huge concern, and it often takes new operators a very long time to build confidence. And then in the ultracentrifugation step itself, As I mentioned with isopycnic on the previous slide, it takes a long time to form the gradient, and so these typically have run times of 24 hours or longer. Finally, recovering the sample is another step that I would say suffers a bit from inconsistency because it is manual. And since most recovery methods utilize needles, there's an inherent safety concern as well. So we've been working hard to develop solutions to these challenges. So automating the preparation process allows us to eliminate variability and afford significantly shorter run times for those linear gradients. And then also by making recovery safer, we can ensure that you can maintain the high purity and yield without sacrificing operator safety. And so this is where OptiMATE comes in. The instrument is designed to reduce error and improve reproducibility by completely automating gradient dispense and sample dispense, as well as tube sealing. So with the ability to dispense those preformed gradients I mentioned, you can cut your spin times by over 75%, allowing you to accelerate your workflow. And it has a really simple user interface that enables new users to begin creating these high quality gradients on day one. And so in many ways, it introduces standardization to your lab. And with a really small footprint, it can fit very easily in biosafety cabinets or directly next to your ultracentrifuge on the benchtop. And really our objective when we set out to develop this instrument was to simplify as many aspects of this workflow as possible. And so as Bala will share with you in some subsequent slides, it really does make life easier for those that are running density gradients. OptiMATE is compatible with the most commonly used gradient materials, so the big three kind of being sucrose, cesium chloride, and iodixanol. And of course, with cesium chloride and iodixanol, we have our validated OptiMATE reagents, so these will work with the instrument, but you're also free to use your own stock solutions. So you would just need to define the density or concentration of those solutions. It also supports a broad range of buffers and additives, as you can see from that list on the bottom. And this list is certainly not exhaustive, but it shows some of the components that we have tested the most rigorously. The system also works with a broad range of Beckman-Coulter rotors and tubes, and these include tubes ranging from smaller scale down to 13.5 mils, all the way up to 94 mil tubes. And it's compatible with a variety of different tube formats as well, including quick seal, OptiSeal, and open top. So whether you're running a small scale prep using something like the Type 90 Ti or a high volume separation with the Type 45 Ti, OptiMATE can integrate pretty seamlessly into your existing ultracentrifuge workflow. And importantly here, I'll play this quick video. OptiMATE doesn't just dispense gradients and sample. In the case of quick seal tubes, it's able to seal them in parallel while dispensing. So this is basically a time lapse of OptiMATE preparing 3 tubes. And so on the screen on the instrument, you can see the countdown running, and it takes roughly 5 minutes per tube if you include sealing. And so the instrument is first going to check whether the components and tubes are set up as expected or as defined in the method, and then it will begin dispensing, as you can see now. And so it fills most of the tube, and then it has a top-off function. And this basically ensures that the tubes are filled completely, so they don't have to be weighed, assuming you have equal components in opposing tubes. And once it moves to dispensing the 3rd tube, as you can see, it now begins to simultaneously seal the first one. So this also eliminates one of the more tedious and variable parts of the workflow to drive consistency, not only from tube to tube, but across replicates, runs, operators. And so now I can hand it over to Bala to discuss some of the experimental work that has been done.
My name is Bala Venkatakrishnan. I am an application scientist at Beckman Coulter Life Sciences, and it's my pleasure to be talking about the significant enhancement that the OptiMATE Gradient Maker brings to density gradient ultracentrifugation workflows. So following up on what Shawn just shared, in this slide and the next few, we will use density gradient workflows of adeno-associated virus, or AAV, the popular gene therapy vector, as a benchmark to test OptiMATE's ability to make the process easier and faster. So typically, an AAV purification workflow will have two steps. The first one to separate capsids from other cellular material when it's in a cell soup or cell lysate format. And then there is a second step to separate viral genome-filled capsids from empty capsids. Now the second step is especially important for gene delivery applications, and that is where cesium chloride density gradient ultracentrifugation is often considered to be a gold standard. So we will walk through this workflow stepwise. The first step, where there is separation of cellular material from AAV capsids, amongst other techniques. A popular technique that utilizes density gradient ultracentrifugation is an iodixanol step gradient. And in this case, it's a 15 through 60% weight by volume concentration iodixanol step gradient. Then we will first describe the current state where you manually set this up. You will either overlay or underlay these various layers of concentrations of iodixanol to create the step gradient, after which you will dispense the sample on top, and that is shown in red here. You manually dispense this, then you seal it, and then you do an ultracentrifugation for about 2 hours at high speed. And then at the 40%, 60% interface, you will be able to pull out your capsid band of interest, which will have enriched folds that's in the space, but you may not always be able to visibly observe these. Now, the ideal state that the OptiMATE Gradient Maker will offer here is the automated dispense and seal of this. So this manual process, as you can imagine, is painful when you have to do more than one tube. It is time consuming. You still have to be careful to make sure that there is no cross diffusion between these layers as you're making them, which can create a bunch of errors if you have untrained hands, or depending on training, there's just a lot of variability that you can expect user to user, which is not desirable. So with the OptiMATE Gradient Maker, there is precision and accuracy that's brought to this workflow, and so it will dispense this in automated fashion, include the sample inside, and then seal the tube if you're using a quick seal tube here. The ultracentrifugation and the rest of the process proceeds the same way. So the key benefits you're looking at here is because of the precision, you see a significant reduction in errors. There is no manual step involved in actual gradient making, so there is lower barrier to entry. As a result, and I will show you some data on this, there is a significant reduced hands-on time, and then you will have improved reproducibility as a result of all of this. As a next step, you do the separation of the full capsids from the empty capsids. And for this, as I had mentioned, the cesium chloride isopycnic gradient is the gold standard. The current method for doing this is you have a mixture of cesium chloride and your AAV sample. It becomes a homogeneous mixture, and once you dispense it into tubes, you seal the tubes, and then you do the ultracentrifugation, which is a rate limiting step. In this case, we have encountered workflows where customers talk about anywhere between 16 hours to 48 hours, sometimes even more, for this separation so that it proceeds to equilibrium. That is the self-ordering or self-forming of the gradient of cesium chloride. And as part of this, you will find that the full capsids and the empty capsids separate into specific bands, after which you can extract the full capsid band using a syringe when you puncture the side of the tube. Now with the OptiMATE Gradient Maker, as you can imagine, the steps involved in the manual dispense have not necessarily been described as laborious and error prone. The key benefit you're looking at is shorter runtime. And I'll talk about the mechanism of this in the next slide in detail. What you do is you create a preformed gradient with defined upper and lower limit densities. And then you do the ultracentrifugation, where under the influence of the ultracentrifugal force, you have the migration of the particles to match their equilibrium densities, and then you actually get this band separation. You can extract the full capsid bands. So there's a significant shortage or significant decrease in the ultracentrifugation runtime as a mechanism, and you will also see some of the other benefits that you will generally see when compared to manual gradients where you have reduced errors. One key factor is you can have accurately computed cesium chloride amounts, especially when you compare this with powdered cesium chloride, where because it's hygroscopic, there is some difficulty in having the right amount added. The other aspect is when you're adding powdered cesium chloride to a solution, the final volume is not always predictable. This allows you to avoid that because you can use everything in solution format. And again, some of the other factors, lower barrier to entry, reduced hands-on time, improved reproducibility is all that is offered by the automation of the OptiMATE Gradient Maker. So how does a preformed gradient do this? This slide will elaborate on that. So in a manual run, what you're looking at is this homogeneous gradient over time taken in the ultracentrifugation process slowly starts ordering, self-forming this gradient. Right? So over time, it'll end up forming this equilibrium format of the gradient. Now, when you already have a preformed gradient, this gradient formation is the rate limiting step. That's the reason why you ultracentrifuge it for so long. But with the OptiMATE system, because you have a preformed gradient, the runtime becomes entirely dependent on the migration of the sample. And so as the sample moves towards the position that matches its particle density, this process takes a lot less time compared to this, where there is say self-forming also occurring. In other words, you've skipped the first step, which is the self-forming of the gradient, and gone ahead and just had particle separation. So with that, from this slide onwards, we will delve into the details of how we actually are able to test this hypothesis for cesium chloride. Before we get into the purification of AAV, though, we will first delve into the details of the tube setup. So here we compare a manual cesium chloride gradient tube generation process and the OptiMATE process. For the manual, you mix a cesium chloride solution with sample. We use water here. And you dispense it, in this case, into 13.5 ml quick seal tubes that will go into a type 70.1 TI rotor. And then you do the sealing manually in the case of the manual. You're going to be doing an ultracentrifugation for 20 hours to allow it to form that equilibrium format gradient. Once that is done, you fractionate the post-run samples from the bottom of the tube by puncturing. So in comparison with the OptiMATE Gradient Maker, it dispenses that preformed gradient that we just talked about with set upper and lower limit densities. We're going to ultracentrifuge it for just 4 hours and then collect the fractions. And what these fractions will enable us to do is generate what we call a gradient profile, which essentially looks at the density of the actual sample through the length of the tube, and then we will use that as a comparison tool. So the data on the slide shows two results: a comparison of time taken, specifically hands-on time in manually dispensing and sealing two 13.5 ml quick seal tubes versus OptiMATE, and then a comparison of variability in the OptiMATE Gradient Maker dispensing a preformed gradient between 3 tubes. So we'll focus on the time comparison first. The manual process involves the mixing of your sample. In this case, we'll just use water with cesium chloride, which is a fairly quick step. You then go ahead and dispense it into 2 tubes. Now, this takes a little longer simply because you're trying to avoid any bubbles that are formed and stay trapped inside of the tube when you're using a syringe with a blunt end cannula. So you still do this a little bit carefully. You can do it faster, I guess, but there are some risks with it. The next step is then the sealing of the two tubes, which again is not a very time-consuming step. So for 2 tubes, you're looking at 14 mils in total. Now with the OptiMATE Gradient Maker, what you do is you initialize the instrument and connect your reagents, your gradient modifier and sample to the tubing. You select the method, you prime the tubing, you calibrate if you need to, and then you go ahead and ask it to dispense and seal. This takes about 7 minutes, but the key thing here is there's no manual intervention. You hit go and you walk away. You can set up your ultracentrifuge or your rotor. And so you're looking at a hands-on time of just 5 minutes. The emphasis here, again, though, with the cesium chloride gradient, is not on the time saving in the prep of the tube, because again, this has not necessarily been described as a laborious method, but it is A, the precision that you achieve, and B, the significant saving in the ultracentrifugation runtime, which we will talk about in the next slide. This figure shows a plot of density on the y-axis and cumulative volume from the top to the bottom of the tube on the x-axis, something we can refer to as a gradient profile, and it shows a comparison of 3 different tubes dispensed by the OptiMATE Gradient Maker. And in this case, I will point out that tube 3 was actually dispensed the next day, and you'll still find that there is excellent overlap between these. And I'll start showing you the same kind of data, but with error bars to show you how accurate it can be. This slide shows you density and purification profiles for an AAV cesium chloride purification from 2 different rotors, accounting for 2 different commonly used tube sizes. 13.5 mil with the type 70.1 TI rotor, and then 39 mils with the type 70 TI rotor. So after ultracentrifugation, the homogeneous cesium chloride solution will form a linear gradient after 20 hours, with some curvature towards the bottom of the tube, a certain nonlinearity, which is typical of cesium chloride gradients if you refer to our rotor manuals. The preformed gradient, which is shown in red, retains a gradient profile that matches the cell-formed gradient after a 4-hour ultracentrifugation up to a density of approximately 1.4 grams per mil, after which the curvature of the cell-forming gradient starts. The preformed gradient remains more linear because of the shorter ultracentrifugation time. We have marked the expected density-based locations of full and empty capsids in both of these gradient profile plots here. But then when we go ahead and use this, so the overlap is nice, but this difference is something that we will explore in a little more detail in this slide and the next slide. But when we purify AAV 9 particles using these density gradient parameters, and in this case we will use a 5-hour ultracentrifugation just so that there is slightly better separation and resolution between the bands. There is still an observable separation between empties and fulls, though it may not exactly match up. And some of that is because of this difference that you observe. And we'll talk about these sort of resolving of the full capsid bands into 2 bands in just a bit. Here you can actually see those 2 bands, though they may not be necessarily recoverable as separate entities. So with the 39 mil tubes in a type 70.1 rotor, you see a similar pattern, both in terms of the gradient profile as well as these additional bands. So the additional bands with the preformed gradient seem to be a consistent observation for us, and in fact with our customers with their samples when they use OptiMATE. So one question is this semi-purification, or are these partially filled capsids? So I'll show you some data in a couple of slides that they're actually a different stoichiometry of full capsids that we're able to resolve because of the decreased steepness and therefore better resolving power that the preformed gradient run for shorter time offers. So Susumu Uchiyama's team at Osaka University has extensively studied these multiple bands, and they call them FP1 and FP2, or full particle 1 and full particle 2. So what they identify is there is actually no difference in the packaged material using this capillary gel electrophoresis data. There is instead a difference in the VP1, VP2, and VP3 stoichiometry, which they quantitate here, where one band population has additional VP1 and VP2 content on average. This they find correlates actually with increased in vitro transduction efficiency, as you would expect, because of the increased presence of the VP1 unique region with the phospholipase A2 domain that enables better trafficking, and the increased number of nuclear localization signals offered by both the VP1 and VP2. So in a sense, what this allows us to conclude is that the entire separated full capsid particle bands are both useful material from a gene therapy perspective. That the payload is really the same. It's just different compositions of VP stoichiometry. So this is not the resolving you see from the preformed gradient is something you don't see in the manual simply because of the curvature, but the additional resolution helps you identify some of these factors. And with AAV, while this is the story, it leads to a larger thought process where having a preformed gradient just gives you generally more resolution, which for more impure material or more heterogeneous material, having more resolution is actually very useful. So these recovered full capsid bands were analyzed by analytical ultracentrifugation for purity, and it was found that there is a very good match between what OptiMATE Gradient Maker generated preformed gradients, ultracentrifuge for just 5 hours. It matches very well with what is offered by full 20-hour ultracentrifugation by self-forming of the density gradient. In all cases, the purity is consistently around 90% across the 2 different tube sizes, 2 different rotors. In the case of the type 70.1, we just are able to use more tubes, so we have some error bars here as well. Just for comparison, you see that for empty capsids that were recovered from the same sample, there's a sharp peak at about 60s and a very minor full capsid peak, whereas our full capsids are all around 80 to 100s with negligible presence of empty capsids. So in all, the OptiMATE's hypothesis of using a preformed gradient for reduced ultracentrifugation runtimes works, and it is able to reduce runtimes by 75% without sacrificing resolution. So to bolster these findings, I also have data from a beta testing by a customer, in this case Capsida Biotherapeutics, where they compared their current manual method to a method ported to OptiMATE with a significantly shorter ultracentrifugation time. So the Capsida team is able to include a lot more AAV, so we have some better band images here. And while I'm unable to share actual parameter numbers, you can see a faint additional band for the full capsid here as well. So they tested their viral genome recovery and find it comparable to their manual method, despite a shorter ultracentrifugation runtime enabled by a preformed gradient. And I'm excited to share this data because it's a testament to what our customers can do when enabled by our technologies. And sometimes it is a lot more innovation that they come up with. They are less conservative than us sometimes and push the frontiers of what the instruments can do. So while again I'm unable to share raw numbers in terms of the run parameters, the Capsida team was able to reduce ultracentrifugation time from what we just claimed, which is about 4x to 5x or 6x or even 10x to their manual ultracentrifugation time. So what you're looking at is, we had a self-forming gradient forming in 20 hours, so a 10x reduction is talking about doing it in 2 hours. This again is with no compromise on purity, as they analyze using 2 different methods here. And this stands as the strongest evidence of what the OptiMATE Gradient Maker is capable of in empowering density gradient ultracentrifugation workflows while doing it easier, faster, and better. So in this final piece of data on cesium chloride gradients, we build off of the data that I've shown you thus far to show that the improvement that we're able to make is scalable. And so we use 2 of our higher capacity ultracentrifugation rotors, the type 50.2, which can take 12 times 39 mil tubes, and the type 45, which is our largest ultracentrifugation rotor, which can take 6 times 94 mil tubes. In the gradient profiles, you can see that we can replicate a significant section of the linear parts of the self-forming gradient, shown in black here, in the manual method by using OptiMATE in a preformed gradient shown in red. There's good overlap, there's very small error bars. There is also the expected separation of bands in the preformed gradient with a clear presence of the two full capsid bands, as you can see in this 94 mil tube. So for this slide, what we will do is we will switch gears to discuss how the OptiMATE Gradient Maker helps with step gradients. In this case, with an iodixonol step gradient that I described previously. And here, again, as I mentioned, the impact is not so much in the ultracentrifugation runtime. It's actually pretty short anyway, but the focus is much more on the effort involved and the quality of these gradients. And similar to cesium chloride linear gradients, we compare here the time taken, specifically hands-on time, and we compare the gradient profiles as well, and look at a little bit of the recovery efficiency by qPCR. So first step, focus on the time taken. So in the manual process, we're using 39 mil tubes here. So preparing the stocks for each one of these layers takes a certain amount of time. And we're not involving any calculation time here, just preparing the stocks. And then you do the dispense of the step gradient, and this is time consuming because you do not want any diffusion occurring. You could probably do this faster, but then there is a risk. And what we want to represent is an average ham trying to do this. So for two 39 mil tubes, it takes about 18 minutes in our hands, and then you seal the tube. So you're looking at a total hands-on time of 31 minutes for 2 tubes. With the OptiMATE Gradient Maker, the steps are the same as I described for the cesium chloride gradient. So you prepare the instrument, you connect the reagents, you select the method, you do the priming of the tubing, calibration if you want to, and then you walk away after hitting the go button. It'll dispense and seal. It takes about 14 minutes, a little bit shorter than this, but in total you're looking at a hands-on time of 5 minutes. And then when we look at the gradient profiles, this is a manual gradient in blue that I dispensed, and in red you see the OptiMATE Gradient Maker dispensed step gradient. And what you see in this faint light gray is what we expect of pipetting errors. And so what you see is an OptiMATE dispensed iodixonol step gradient matches very well with the manual gradient. I wish I'd made more errors here, but you could make something of sharper interfaces coming out of the OptiMATE dispense gradient, whereas the manual dispense is a little more diffuse. Overall, though, the step gradient looks like a step gradient, the way it is meant to be. And once we recover samples after ultracentrifugation at the 4060 interface, we look at just the efficiency of viral genome recovery, and that stands as around 39% with both the manual as well as the OptiMATE Gradient Maker method. And this number also stays fairly in line with what we would expect from my first step purification of AAV capsids. So in this last slide, I want to share a glimpse of what we have on the horizon. The OptiExtract tool to address a key pain point in sample recovery that Shawn talked about earlier in the webinar. So the OptiExtract is a simple tool for the mounting, elimination, and syringe injection into the ultracentrifugation tube for extracting your bands. And so what you will do is you'll take your tube, you will mount it using these brackets. These brackets are different for different tube sizes. You are able to measure the band position using the scale for consistency. And then this dial allows you to adjust the white light below this, so you can illuminate your bands. This moving stand is the key part of it, and this accepts the syringes. You can use one needle for venting, And so you see others for extracting the bands, and you move by pushing your thumbs into the back of the bottom part of the stand. So there's no jerky and clumsy back and forth or twisting of your syringes depending on your tube type. You go all in one clean push and completely eliminate any safety concerns that could occur with the extraction process. Now, something that I am guilty of when I'm trying to position my syringes or needles to puncture into the tube is I will involuntarily have my hand reach out to the back of the tube to try to sort of position myself effectively, give myself leverage. Now you do not need to do that because this goes in one quick, non-clumsy motion. So it takes away that safety concern and overall can help you improve your consistency.
Thank you, Bala. So just to put a ribbon on this slide summarizes how the OptiMATE Gradient Maker affords significant improvements for sort of the current state cesium chloride protocols where you're using linear gradients. So generally, stock of gradient materials are prepared and mixed with samples from upstream processing, and then tubes are sealed and loaded into a rotor. Then you do the ultracentrifugation step where you're using a considerable amount of time here. And once separation is achieved, you stop the run and recover your sample. So there's numerous steps throughout this process that are highly manual, which is indicated by the exclamation points. And overall, you're looking at a process time of roughly 2 to 3 days and training taking anywhere from weeks to even months. But with OptiMATE, you are simply creating a method, connecting your accessories and consumables like our OptiMATE reagents, and clicking start. So the instrument will actually do all of the diluting and mixing for you, allowing you to form this linear gradient before even beginning the centrifugation step. And that can reduce your runtime down to a single working shift. And because most of the key steps are automated now, new users can be trained in just a few hours. And as already discussed before, OptiMATE can be integrated into your existing workflow pretty seamlessly because it enables the use of already established protocols. And so this brings a certain level of standardization and huge runtime savings and consistency to your lab. So that brings us to the end of the presentation. Much of the data that Bala discussed today can be found on our website in application note format. So please scan the QR code to download a copy. Thank you all for your attention, and we're happy to stick around and take any questions.